Alright, hi everyone. So this is the first video for the metabolism chapter. So in this video we're going to go over some kind of terminology and important things you're going to need to know for the process of metabolism, and then we'll go through the actual process of aerobic respiration. So then in the second video we'll review that process and then talk about how things other than glucose are used for energy. Okay, so let's go ahead and dive right in. All right, so for our terminology overview, a lot of this will be review, and then we'll be adding in a few kind of new concepts as well. Okay, so first off, with metabolism, we're going to be forming bonds and we're going to be breaking bonds. And so anabolism is just a term for forming bonds. So if we have, for example, two glucose molecules, all right, and we link them together to form a disaccharide, all right, that would be an example of anabolism. And so that would be done through dehydration synthesis. So we'd remove a water from those to form that bond. Okay, and so then catabolism would be the opposite. So we would just take that disaccharide and we would split it apart to give us two separate glucose molecules. All right, and so that would be done through hydrolysis. So you would add water to break that bond. Okay, so then last chapter we did a review of the different nutrients. So let's just kind of recap them again. And this time talk a little bit about their functions and why they're important in the body. So nutrients, by definition, are things that are absorbed from food that can be used by the body. Our first category is carbohydrates. And so carbohydrates are used for energy, all right? So glucose is primarily what we're going to be talking about with aerobic respiration, being used for energy. They're also used for structure within a cell. So glycoproteins and glycolipids on the surface of the cell, and within the inside of the cell you can have some starches, right, giving structure to a cell. Okay. Then we have lipids. So lipids obviously are a way of storing energy, so you can store fats for energy. Lipids can also be used to make hormones. So there are quite a few fat-soluble hormones, and those are going to be made from generally the essential fats like omega fatty acids and that kind of thing for hormones. And then membrane lipids. So we know the cell membranes are made up of phospholipids, and there's also cholesterol in that membrane, so you need a lot of fats for that as well. And then we have proteins. And so proteins have multiple functions. They can be used for energy, which we'll talk about in the next video. They're structural, so things like collagen and keratin and microtubules within cells are all made up of proteins. And then we know that enzymes are proteins, right? And enzymes serve an extremely important function in the body, right? Carrying out the different processes and catalyzing reactions. Okay, so also with reference to proteins, when they're being used for energy, we talk about nitrogen balance. All right, so this is just the kind of status of protein metabolism in the body, right? So how much are proteins being broken down in the body? Okay, so if you have a positive nitrogen balance, that means protein anabolism is going on. All right, so we're building proteins, putting them together. So we're going to have less nitrogen waste in that case. All right, and so then if you have a negative nitrogen balance, proteins would be broken down, and that would increase the amount of nitrogen waste. And we'll talk about what that nitrogen waste is in the second video. Okay. And then finally, we have kind of our last three categories. So vitamins, we talked about in the last chapter, those fat-soluble vitamins, the CADE vitamins, vitamin K, A, D, and E. And there's also water-soluble vitamins, like B vitamins and C vitamins. Then there's lots of minerals, sodium, potassium, calcium. It's also really important to get iron from the food you eat to make hemoglobin, and magnesium, which is really important for a lot of enzymes to be active. They need magnesium. And then water, we know, is absorbed from food in the small intestine, the majority of it in the small intestine, 90%, and then 9% in the large intestine. And it's used for many processes, including cellular respiration, not cellular respiration. We make water in cellular respiration, sorry. It's used for hydrolysis. So chemical reactions, when we're breaking things down, we're adding water to them for hydrolysis. Okay, so those are the nutrients from food. So with metabolism, the point of metabolism is to make ATP, and ATP is what we use for energy. Okay, so it's the source of energy for many different reactions in the cell. All right, and ATP stands for adenosine triphosphate. Right, and when you look at kind of a schematic drawing of ATP, we would have the adenosine part here, just represented as an A, and then tri, meaning 3, we're going to have 3 phosphates attached. All right, and what's important is actually the last phosphate, and so it's attached by a special bond called a high energy bond. So there's a lot of stored potential energy in that bond. And so when you break that bond, all right, and you take ATP, all right, with 3 phosphates to ADP, which only has 2 phosphates, and then you have that free phosphate, and you just release that energy, which can then be used for any number of things within the cell. Okay, so you're going to break that bond to release the energy that's stored in it. Okay, now this next section is going to be new information, and it's going to be very important for aerobic respiration and understanding the process. Okay, and so this is going to be a new category of molecules called coenzymes. And so coenzymes are molecules that are required for enzyme activity. Okay, so they kind of help enzymes do their job. All right, and our first coenzyme is NAD. And so NAD is derived from vitamin B3, niacin, and it's going to be involved in these reactions called reduction and oxidation reactions. And so that just means, so a reduction reaction means that a molecule is gaining an electron, or in this case, we're always going to be adding hydrogens for our reduction reactions. Okay, so but more generally, it's referring to adding of an electron. We're going to use hydrogen, though, as our specific atom or element. Okay, and then oxidation is losing an electron or a hydrogen. Okay, and so one way I think it's easy to remember this is there is a mnemonic for this. So oil and then rig. So you can kind of remember which is which. And so what that stands for is oxidation is loss, right? Because you're losing an electron, and reduction is gain. Okay, because you're gaining an electron. All right, and that's always how I remember it, oil rig. So oxidation is loss, reduction is gain. So you're gaining an electron or a hydrogen with reduction, and you're losing one with oxidation. Okay, so our example with our NAD, what it can do is it can pick up a hydrogen. So if we take NAD and then we have it bind to a hydrogen, it becomes NADH. Okay, so going to the right, because this reaction can go either way. All right, so going to the right, taking NAD, adding an H to it, and making it into NADH is going to be a reduction reaction. Okay, and if we reverse that and remove the hydrogen, that's an oxidation reaction. Okay? Okay, so NAD can be involved in reduction and oxidation, just depending on which state it's in. All right, so that's our first coenzyme. Its job is to pick up hydrogens and carry them to later parts in our aerobic respiration. All right, so our next coenzyme is FAD. All right, FAD comes from vitamin B2, riboflavin. And so FAD can bind to a hydrogen, actually 2 hydrogens, and become FADH2. All right, so we go from FAD to FADH2. So going this way, again, reduction. We can go back and turn it back into FAD, and that would be oxidation. All right. So both NAD and FAD can pick up hydrogens. Now, coenzyme A is a little different. It doesn't pick up hydrogens. All right, we abbreviate it usually CoA. It comes from pantothenic acid. And so what it actually can bind is an acetyl group. Okay, and so an acetyl group is a 2-carbon molecule. Okay, and that'll be important kind of as we go through the steps of cellular respiration. We'll talk about kind of how many carbons we have in each molecule we're looking at. And so when we get to a 2-carbon molecule, that will be our acetyl, and that can bind to CoA, and it can become acetyl-CoA. Okay, and so then that CoA just functions to carry that acetyl group to the next step of our process in aerobic respiration. All right, so those are our coenzymes. We have 3, NAD, FAD, and coenzyme A. Okay, so now let's talk about glucose. So this will be our last topic before we dive into the steps. So glucose, we know it's a monosaccharide. All right, the chemical formula for glucose is C6H12O6. All right, and so if we're just looking at that, so it has 6 carbons. All right, it has 12 hydrogens, and so remember the NAD and FAD, their job is to carry hydrogens to the last step of aerobic respiration. And actually it is those hydrogens that are being carried to the last step that is going to be used for energy. So it's really these hydrogens, these 12 hydrogens, that are going to be used to make ATP. Okay, they're going to be that energy source, and we'll see that in the process when we look at it. All right, and it also has 6 oxygens. Okay. It's absorbed in the small intestine, and so when it's absorbed, insulin is actually going to function in that absorption, right? And it actually allows glucose to enter the glucose transporter and go into a body cell. And so what insulin does, essentially, it's like the key, right, that unlocks this little door here, right, on our glucose transporter, and then that way glucose can get into the cell. So once glucose is in the cell, it actually will have a phosphate added to it, and it becomes glucose 6-phosphate. All right, and that phosphate just traps it within the cell, so then it can't leave the cell, and so that way that cell can store that glucose. Okay, and so once that glucose is in the cell, it can be converted into glycogen. All right, that process is called glycogenesis. All right, making glucose into glycogen. And then when your cell needs that glucose for some purpose, all right, it can break down that glycogen. That's called glycolysis. All right, and no, okay, yeah, I didn't think that was the right word. Hold on, it's not glycolysis. It's glycogenolysis. I'm going to say glycolysis is the first step of aerobic respiration. All right, so glycogenolysis is the breaking down of glycogen, all right, to glucose when you need glucose. So then that glucose that you've freed up, right, from the glycogen can now go into our first step here of respiration, right, whether it's anaerobic, or aerobic. You'll take that glucose and use it for the first step. Okay, So let's start talking about our steps of breaking down glucose to make ATP. So we're going to first talk about anaerobic respiration because it's very simple, all right? And the first step, right, really the only step for anaerobic respiration is the same exact first step for aerobic respiration. Okay, so anaerobic respiration occurs when there's no oxygen present, all right, and it yields only 2 ATP for every glucose that you put in. And this process is reversible, all right? So actually, to be accurate, right, we would add double arrows on these, okay? Because it can go both directions. Okay, so this first step is called glycolysis. And glycolysis occurs in the cytoplasm. And what's going to happen is we're going to take our glucose, all right? So this is just a drawing of our glucose. It has 6 carbons, like we said, C6. All right, we're going to break it essentially, and it's a little more complicated than this, but for what you guys need to know, we're essentially going to break it in half. Okay, and that'll give us 2 molecules called pyruvic acid or pyruvate. Okay, and so each pyruvate, if we're breaking our glucose in half, it had 6 carbons. Each one is going to have 3 carbons. Okay, so pyruvate or pyruvic acid are 3 carbon molecules. So we're going to have now 2 3-carbon molecules because we divided our glucose in half. Through that process of breaking down that glucose, we're also going to make 2 ATP and 2 NADH. All right, so this is glycolysis, okay? However, if there's no oxygen present in the body, we're gonna finish kind of the process by converting these pyruvic acids, these pyruvate, 2 of them, into lactic acid. And so then that process requires the use of those NADHs that were made. Okay, so those NADHs are used to convert the pyruvic acid into lactic acid. So then all we end up with, our final products are going to be that ATP from here and that lactic acid that we just made here. Okay, so we convert our pyruvic acid into lactic acid. That's going to become a waste product that can be gotten rid of from the body, and 2 ATP, which are going to be the energy produced by anaerobic respiration. Okay, so that's anaerobic respiration when there's no oxygen. So now let's go through aerobic respiration. So aerobic respiration is going to occur when there is oxygen present. All right, aside from glycolysis, the remainder of the steps are going to occur within the mitochondria. Okay, so just to recap what's going on with glycolysis we just talked about. So we have our glucose molecule. All right, so this would be glucose. We're going to divide it in half, and that is going to give us our 2 pyruvate. Okay, and also our ATP, 2 ATP, 2 NADH. Okay, so we're going to have 2 pyruvate and those 2 ATP and the 2 NADH. Okay, and so kind of keep track of, and that's why I'm writing them in these different colors, how many ATP and how many NADH are being made in each step. Okay, so then for the next step, all right, since we're already kind of familiar with glycolysis from our anaerobic respiration, our next step is the formation of acetyl-CoA. All right, and so for this, we're going to use these pyruvates that we made in our glycolysis. Okay, and so this step occurs in the matrix of the mitochondria. And so if you remember kind of the structure of mitochondria, it's kind of got like this oblong shape like this with the separate inner membrane that's very wavy. All right, so the matrix is this inner part, right? It's the fluid on the very inside of the mitochondria. And then you actually have that wavy inner membrane that forms what's called the cristae. Okay, so the matrix is the inner part. That's where this step is happening. And so what's going to happen is we're going to take our pyruvic acid. Remember, we have 2 of them. All right, we're going to start to kind of break it down. And so for your exam, you don't really have to, you're not going to have to draw chemical structures or anything like that, but you need to understand kind of what's going on here, okay? So let's kind of color match these, all right? So our first step, what we're going to do is we're going to take this hydrogen off here, all right? We're going to take that hydrogen and we are going to add it to an NAD to form NADH. And now remember, we have 2 of these pyruvic acids. For everything we're going to talk about is going to happen twice. So we're going to make a total of 2 NADH. Okay? And so then the next step, we are going to remove, let me kind of actually adjust this a little. Make this a little box. There we go. Okay, now we're going to move this oxygen, carbon, and another oxygen. Okay, and so that is going to be taken off of that pyruvate molecule, and that is going to be carbon dioxide. All right, and that carbon dioxide is then just going to be exhaled. Okay, and so for this reason, because we remove a carbon dioxide from this molecule, which is a process called decarboxylation, removing carbon dioxide, and then that is exhaled, this process is irreversible. Okay, because once you've exhaled that carbon dioxide, you can't get it back. All right, so it's irreversible. So this step, taking that pyruvate and converting it into acetyl-CoA, which is what we're doing, is irreversible. All right, so up to this point, we've made 2 NADHs and 2 carbon dioxide. Those carbon dioxide are exhaled, and then we're going to hang on to those NADHs for later because we're going to use them at the end. All right, so now what we're left with, you may remember from when we talked about our coenzyme A, is this 2 carbon molecule here, okay? And so this 2 carbon molecule is an acetyl, okay? So that acetyl can be added to CoA to give us 2 acetyl-CoAs, all right? So from the formation, this step called the formation of acetyl-CoA, we're gonna get 2 NADH, 2 carbon dioxide, they're going to be exhaled, and 2 acetyl-CoAs. All right, those are our products. And we started with pyruvic acid, 2 pyruvic acids. All right, so now let's talk about the next step. So the next step is called the Krebs cycle, also sometimes referred to, just depending on what you're looking at, the citric acid cycle. It's referring to the same thing. This is going to be also occurring in the matrix of the mitochondria, so that kind of inner, in this picture, more orangey part here is where it's happening. This is a cycle, okay, so it's going to be kind of a process that's happening kind of in this circular format. There are many, many, many steps in the citric acid cycle, and I don't want you guys to worry about any of those specific steps. What you need to know is what goes into the citric acid cycle and what do we get out of the citric acid cycle. Okay, so what we're going to put into the citric acid cycle are our acetyl-CoAs. Okay, so we had 2 of them that we made in the last step. So we have our acetyl-CoAs, 2 of them. We're going to put those in, and then for every acetyl-CoA, we are going to get 3 NADHs, so that gives us 6 total because we have 2 acetyl-CoAs. We're going to get 1 FADH2, so that gives us 2, all right, because you get 1 for each acetyl-CoA. You get 2 CO2s, which makes sense, right, because we have 2 carbons here, so that would be used to make 1 carbon dioxide, that would be used to make another carbon dioxide. So each acetyl-CoA can give us 2, so that's 4 total. and then we make 2 ATP. All right, so again, the CO2 would be exhaled. All right, so this step also is irreversible. You can't go backwards and, you know, put these things together to make an acetyl-CoA. All right, you can only go forward for this. Okay, so then if we go through after this step and we tally up and get our net total of our coenzymes, our NADH and our FADH2, we have 10 NADHs. So let's double check that. All right, so if we go back to the top, we have 2 from here, our very first step in glycolysis. We added 2 more with the formation of acetyl-CoA, that's 4. And then we added 6 here, so that's 10. Okay, good. So we made 10 NADH. And then this last step with our Krebs cycle is the only place we've made FADH2, and so we only made 2 there. And our ATP, we've made 4 total, so let's double check that. So our first step, glycolysis, we made 2 here. We didn't make any with the formation of acetyl-CoA, but we did make 2 here in our Krebs cycle, so that's 4 total. Okay, so this is our kind of running total here of things that we've made so far. All right, so now we're going to go into our very last step. So this is where we're going to use all of those coenzymes that are carrying those hydrogens for us to make ATP. Okay, so this last step is called the electron transport chain, and it occurs on the cristae of the mitochondria, so this folded membrane of the mitochondria. All right, and so I usually like to use an analogy for this process. If you were to take a much more in-depth class like biochemistry or something, you would learn all the specific enzymes and channels involved, but I just want you guys to kind of understand generally what's going on. So we're gonna break it down to use kind of the analogy, but I'm gonna briefly explain a little more of the specifics so you kind of understand what the analogy is referring to. Okay, so it's happening on the cristae of the mitochondria, and the way that our hydrogens here that are being carried are used for energy is that they are going to, essentially we're going to use kind of a waterfall analogy. They're going to pass over each waterfall. There's going to be 3 total. They're actually going to be an analogy for a channel in the membrane. Okay, so we would have hydrogen. Let me kind of switch colors here so it's a little more visible. So we're going to have hydrogens, all right, passing through each of these channels. All right, we're going to call them waterfalls. And as they do, they're going to build up enough energy, all right, because we have all these hydrogens now out here. They're going to build up enough energy each time they do that to make 1 ATP, okay? So as the hydrogens pass through each waterfall or channel, it will generate enough energy to make 1 ATP. Okay, so let's look at our analogy. Okay, so here's our waterfalls. So we have our 3 waterfalls. We have our hydrogens. So we said, as our hydrogens pass over each waterfall, all right, they're going to make an ATP. So we would make an ATP here. All right, pass over the second waterfall, make an ATP here and here. Okay, so each time they pass over that waterfall, which is, you know, essentially them going through an ion channel, they're going to generate enough energy to make an ATP. So if they pass over all of them or through each of the channels, they're going to make 3 ATPs. All right, so let's bring our coenzymes into this. Okay, so our NADH is going to come in here, and it is going to drop off its hydrogen right at the very top of these waterfalls. Okay, and so then that is going to turn this into just NAD, right, because it no longer has its hydrogen. So this would have been a reduction reaction. All right, so that NADH was reduced, and now it actually, that NAD can just go back and be reused. So it can go back and pick up another hydrogen from another step. Okay, so then that means that that hydrogen is going to be able to go over 3 times and make 3 ATPs. Okay, so that's what we see here. So each NADH can make 3 ATPs. All right, and we can add that up here. So this makes 3 ATP. Okay, so then we have our FADH2. Okay, so that FADH2 is going to drop off its hydrogen. Let's do that. There we go. Here on the second step. Okay, and so then we'll end up with our FAD, which just like NAD can be reused again. Okay, and so every FADH2 can make 2 ATP. All right, so let's talk about the last step of this electron transport chain. So these hydrogens are passed down the waterfall. Each time it goes, it generates enough energy to make 1 ATP. All right, so now we need an enzyme that is going to use that energy we're generating to actually make the ATP. That enzyme is called ATPase. You absolutely need to know the name of that enzyme. So an ATPase takes the energy from the electron transport chain and makes ATP. Okay, and it actually is this big, if we look back up at our picture up here, right, this big channel that opens up, and it allows all of those hydrogens that are now out here to come rushing back in. And that energy allows it to make ATP. Okay, so this guy is the purple one was our ATPase. Okay, so those hydrogens that have been falling and building up energy as they go pass through that ATPase, kind of releasing all that energy they're storing up, and make ATP. All right, so this process we would take ADP and phosphate here, and this enzyme would make that ATP from it. Okay, so that's the ATPase. All right, so let's talk about our total of ATP that we made. Oh, one last thing. Almost forgot, this hydrogen. All right, so we've ended our process. We've allowed all this hydrogen to rush into the matrix of the mitochondria, which is going to make it very, very acidic. Okay, so we have to neutralize that acidic environment, and that is done by oxygen. All right, the entire reason we breathe oxygen is for this process, so that O2 can swoop in and bind to that hydrogen at the end of this step and make water. Okay, so that's the reason we breathe oxygen. The oxygen is carried by our red blood cells in order to be able to catch this hydrogen at the end of this step and make water from it. All right, and that's one reason. So cyanide, which is a poison, right, you know, the reason cyanide is so poisonous is that it actually blocks this step, right? So it prevents this from happening. And so oxygen can never bind to hydrogen, and so then we end up backing up all of these steps, right? And so your mitochondria end up not being able to make ATP, and then that becomes deadly, right? Because many organs like the heart and the brain require a constant supply of ATP to function. Okay, so now let's talk about our total. All right, so our NADH, if we make 3 from each NADH, and we said at the end of our last step here, we made 10 total, all right, and 2 total FADH2s. Okay, so 10 times 3 gives us 30, and then, whoops, that math doesn't even make sense there. There we go. All right, and then we have 2 FADH2s, each one making 2 ATPs, so that would give us 4 total. So just from the electron transport chain, all right, So I'm going to clarify that. From the electron transport chain alone, so if that's asked on a test question, how many ATP are made in the electron transport chain? It makes 34, all right? So this would be, we get 34 ATP in the electron transport chain from one original glucose that we started with, okay? And so then remember, we also made 4 ATP in our previous steps, so we can add that to our final total and get 38. All right, so we get 38 total from aerobic respiration. Okay, so that's our total for the whole process. Okay, all right. So I know that's a lot, okay? So you should review this and, take your quiz. And then we are in the next video gonna go over and kind of do a summary recap of all of the steps, which I think is a really good way to kind of practice it and study it. We'll take out a lot of these pictures, right? And we'll just go through in each step, what did you start with and what did you end with, okay? That'll be the next video.